Let's see if we can get the technology to work today. Hopefully it actually will. Wanted to briefly finish up with coronaviruses because we didn't get to the fun parts last time because, well, the most fun parts last time anyway. We ran out of time because of all of my technical issues. So today I did want to very briefly just talk about what I think is a much more concerning aspect about coronaviruses and coronavirus disease before we shift over and start to talk about measles and why vaccines are such a wonderful thing. So last time we talked a little bit about SARS, severe acute respiratory syndrome. That came up 2003 and then was rather rapidly gone, probably about 8,000 cases, about 800 deaths, about 10% mortality. People got really up in arms about this. Very few people, however, know about MERS. MERS, Middle East respiratory syndrome, is much more of an issue, but I guess because there haven't been as many cases here, it hasn't gotten anywhere near as much press. So MERS coronavirus, the first indications of this were in 2012, Middle East, and then also some cases in the United Kingdom. 30 to 40% death rate, so 3 to 4 times higher than for SARS. How many of us heard about it? Was it all over the newspapers everywhere? No. However, if you were in Saudi Arabia, it was, because that's really been the major center of this. Index case, we haven't really talked too much about index cases before. This is more of an epidemiological term or an outbreak term. The first case of where someone was diagnosed with this disease was in September of 2012. And then it's been going on for a number of years, again, mostly in the Middle East, partly why the Middle East respiratory syndrome. And as of, this is all totals now, about 2,400 cases, 800 of those are fatal. So still very high rate of mortality for this particular coronavirus, probably partly because of the fact that we figured out that SARS was a coronavirus. People were looking for coronaviruses, and this particular one was also isolated in Rotterdam. And in fact, quite soon after the discovery of the disease, complete sequence, so between the first case and the complete genome sequence was about 6 months, which also tells us that we're doing a pretty good job even with some of these emerging diseases. And it became really obvious that this was a coronavirus. Also, strangely enough, closely related to bat coronaviruses. And again, this should be no surprise based on what we talked about last time about the huge diversity of coronaviruses in bats. And so, even though, as we'll see in just a second here, people are also talking a lot about camels, which again we'll get to a little bit more detail in just a second, is probably a reservoir in bats, and then is transferred via camels to humans, and potentially also that was what was going on with the civet cats in the markets in terms of SARS and the emergence of SARS as well. Just as a quick reminder, this is a classic coronavirus, also interacts with a peptidase receptor. So these peptidase receptors, let's see, who are we at now? Who am I going to put up? Christian, do you remember why these peptidase receptors are potentially so useful for coronaviruses? Here, I'll throw you the virus, it'll help. You want to throw it to someone else? Who else would like to answer why peptidase receptors are so useful for coronaviruses? Diana? To some extent. They're not that common on different cell surfaces. Any other ideas? Anyone else want the virus? What about virions? What's one of the really fascinating things about virions? They've got this metastable state, right? Could be stable on the outside, unstable on the inside. Echo has an idea. Throw the virus.
It uses the peptidase. It
Yeah, so the idea, now this is not completely proven, but the idea is that if you have a peptidase that you bind to get inside a cell, that will also help to break down the virion. And so that's a way of helping genome entry. And so that's one of the reasons that people think that this makes a lot of sense. So that being said, again, classic coronaviruses, mostly in the Middle East. And interestingly, this is just some data, they should say these are some data from the WHO, looking at various outbreaks. The color coding here, dark blue is Saudi Arabia. So you can see that's really the vast majority of cases are in Saudi Arabia. But there's this one blip right here in 2015, the Republic of Korea. And this is South Korea, where they had a single person who came into South Korea in May of 2015, ended up with 186 cases, most of them in hospitals, and 36 of those people died. And as hopefully most of you know, this is one of the main medical centers in Seoul. And so it was really kind of frightening that these are such high fatality rates with some of these diseases. So fortunately, they were able to, mostly through quarantine, actually. It turns out that the way that MERS is spread is through very close contact. And as long as that close contact is avoided, unfortunately, medical care usually depends on close contact, that was taken care of. And just a few months later, that was gone. Why haven't we heard about it? Probably because there haven't been very many cases in the US. Two, both healthcare workers from Saudi Arabia who got really sick really quickly took themselves to the hospital because in Saudi Arabia they knew about this, were immediately tested for MERS, were found to be positive, and there's no good therapies for these. Just had them stay in the hospital and be covered for a while, and then they were fine. But given those two cases, there were almost 1,000 people who were tested for MERS and found then to be negative. So that's where the state is, and again, probably why not all these people have heard about this coronaviral disease that has a 20 to 30% death rate. Where does, I should say, MERS come from? I mentioned Saudi Arabia. Because of it really a focus in Saudi Arabia, people have thought about, okay, where could it be coming from? And they found very quickly that people who were coming down with MERS very often had close contact with camels. And I do not know the culture in Saudi Arabia, but apparently a lot of the camel workers, people have camels as pets. They also have racing camels and are in very close proximity to the people they are working with. And they've gone back, they've looked at these camels for over 20 years, so well before the first, at least diagnosed cases of MERS. Clearly, MERS has been in camels for that period of time. There are some vaccines under development, actually mostly for camels rather than for humans. The numbers here, just a couple of thousand cases, so it's still a relatively low number of cases, but a certain pretty high proportion of the camels do seem to be actually exposed to disease. And it has an almost identical sequence to the human sequences, and so it's probably pretty clearly the connection. The camels seem to get a little bit sick, but not completely. What's a little strange is that people have found that there's MERS in camels in a lot of other parts of the world, but don't seem to be being spread to humans. And so a lot of this actually may be social. Apparently in Saudi Arabia, people get more up close and personal with their camels than they do in other parts of the world. So again, just a guess here. And there's a nice TWIV that talks about this in quite a lot of detail in terms of MERS in camels. So just as an overview, this is where the vast majority of cases are, again, in Saudi Arabia. Some of them imported. Those are those two that came into the United States here, originally found in the UK. And it is possible, actually I should have mentioned this, to get human-to-human transmission of the disease, but only, again, if you're in really close contacts. And it basically seems to be there'll be a couple of close contacts that get the disease, and then probably there are actually other people who are exposed and actually aren't coming down with the disease. They just never get infected enough to be detectable. But where we have the main transmission is really going on here in Saudi Arabia. So probably what's happening goes from camel to camel. In Saudi Arabia, it'll go to humans, and then occasionally those humans will spread the disease elsewhere, which is another reason why vaccinating the camels is probably the best way to get rid of this particular disease. So that was all that I wanted to talk about as far as coronaviruses are concerned. Any more questions on the coronaviruses? Yeah. Yeah, so the question is, what basically, what's the yellow? And so the yellow is where they've done testing of camels, and you do see camels with MERS in those locations. Now, what I don't know is where the bat coronavirus that corresponds to this circulates. That's a more open question that I do not know the answer to. Okay, so everybody's happy with coronaviruses? Which means you should be grabbing your clickers because David's already noticed that. So our clicker question for today on coronaviruses, the structural proteins, I'm going to stop doing this. For in MERS are encoded at the 5' end of the genome, the 3' end of the genome, at both ends of the genome, the middle of the genome, spread throughout the genome. And any of you who've looked at old exam questions know that I love this kind of question. 10, 5. Looks like nobody else is voting anymore. Okay, a couple of things as far as a question like this that might show up on an exam. Couple of things to think about. First, of course, is that MERS is a coronavirus. So immediately, coronavirus have a particular way their genomes are set up. And the sort of unique thing, at least as far as viral genomes that we've talked about so far, is that they have these nested subgenomic RNAs. And those nested subgenomic RNAs are the ones which are encoding for the structural proteins. You need way more of the structural proteins than you do of your non-structural proteins. And that nested, being that we're a positive strand genome, are all going to be present down at the 3' end of the genome sequence. So that makes sense? Yes? Happy? No? Okay, so B is correct. Okay, so now my sort of classic line here as I go and stand over on the other side of the room, because now we're talking about negative strand viruses as opposed to positive strand viruses. These, for the next 3 lectures, we'll talk about these negative strand RNA viruses. So the Paramyxo-Rhabdoviruses, the filoviruses, and the orthomyxoviruses, also known as flu. And when we originally talked about those negative strand RNA viruses at the beginning of the term, we said these are the nasties. And these are definitely some of the nasties: measles, mumps, RSV, rabies. The 2 sort of lab rats of these are the Sendai virus and vesicular stomatitis virus, because they cause a lot less in terms of disease than some of these other ones. MM, and actually rubella, it's not this R here, is one of the best vaccines, and we'll talk about that at the end today if we get a chance to chat about that as well. So what are some of the key concepts here? One of the reasons I asked that question about MERS and subgenomic RNAs, because these viruses also have subgenomic RNAs which are made, but since they're negative strands, it's a very different way that those subgenomic RNAs are made. Instead of doing this nested subgenomic, they have this process they call start-stop, which is literally where the RNA-dependent RNA polymerase will start to make a messenger RNA and then stop and think about it for a while and then decide if it's going to start to make the next one or not. So a very different way that you get messenger RNAs. The other thing that happens in these viruses is they do something called stuttering, which means that the RNA-dependent RNA polymerase just sits in one place and adds one nucleotide, and one nucleotide in particular. So we could toss the virion back to Janan. What particular nucleotide would you really like to make many, many copies of if you're trying to make a messenger RNA? Right in the back, all the way back. And toss it to the virion. Up. So if you're making messenger RNAs and you want to make a whole bunch of one particular nucleotide, which one would that be? The start codon, but that's one part, one end. What about the other end, the RNA? What is usually present in messenger RNAs? You can, but I think you know it. What do you think? So you got the 5' end, what's got a cap. The 3' end has a tail. What's those tail usually made of? Adenines, right. So what might these things be adding lots of? Adenines, exactly. So many, many, many adenines get added through this stuttering process. There's also a process called alternative start codons, which this should sound somewhat familiar anyway. It gives you overlapping open reading frames. So it's not just these tiny little single-stranded DNA viruses that you find in bacteria, but also in some of these negative strand viruses as well. And in a few cases, there's even some RNA editing that takes place. And we talked a little bit about RNA editing in the molecular biology class last term. Turns out that lots of viruses also do some RNA editing processes. So I'll start out a little bit talking about the origin and some of these different viruses. Then we'll go through structure, binding, and entry. But it's really mostly about this replication and particularly the formation of messenger RNAs. So I probably should have made these guys red on our slides here. How they get out is pretty straightforward. These are enveloped viruses. And then at the end, I can wax poetic about why everybody should be vaccinated and why I'm actually thinking about getting another measles vaccine in the near future. So these are, as I mentioned on the other side of the screen now, negative strand RNA viruses. And once again, the virologists have come up with these really incredibly creative names, MonoNegaviruses. So single genome segment negative strand. So these are packaging a single negative strand, as we'll talk about when we talk about the orthomyxoviruses for flu. Those have multiple segments which are being used. They all bud at the plasma membrane, so instead of internally like the Golgi that we had with the coronaviruses, these are all budding actually at the plasma membrane. One really important aspect about these negative strand RNA viruses, negative strands, just sort of by definition, can't be used as something to translate because that's our definition of negative strands. So you have to bring a polymerase with you when you have an infection process, because that's the only way you can make message that can get translated. So very, very different than these positive strand RNA viruses that we've talked about so far. And then what that means is that these RDRPs, RNA-dependent RNA polymerases, are all present in the virion. And come in with that infection process, and I mentioned the start-stop already here. So what are the major ones of these? Rhabdoviruses, these are, and I should have pointed this out, maybe we'll back up here real quick. These are your typical rhabdoviruses down here. People talk about the virions as having kind of a bullet shape. These are best known as the rabies viruses. And so rabies is really actually almost 100% fatal disease if you get rabies. Fortunately, you can have post-exposure prophylaxis because it takes quite a long time for the rabies virus actually to get into your central nervous system where it causes encephalitis. And I think there have been like 2 or 3 reported cases of where people have gotten rabies and then not gotten the vaccine and still survived. So it's a very, very low frequency. And partly because of that, it was one of the very first viruses for which a vaccine was made, and that was through Louis Pasteur. And this is a classic attenuated vaccine. And he started out with rabbits, where you have the red hypodermic here, which represents the nasty virus, and then just took this from one rabbit, put it in the next one, put it in the next one, put it in the next one, many, many, many times, and eventually that virus becomes attenuated. And this was the very first process. And I don't know what that attenuation process did in terms of changes in the virus genome. I'm sure it's known. But it is actually a very, very safe vaccine. And also, again, it works well in a post-exposure prophylaxis. Because, whenever you get infected by rabies, it does cause this encephalitis. Again, if you don't get treatment, not very many people work with wild-type rabies, but vesicular stomatitis virus turns out to be a related rhabdovirus, which is not so good for cows, but for humans seems to cause no problem whatsoever. It also has an extremely wide host range. So rabies has a relatively constrained host range in terms of some different mammals, like certainly rodents, but I forget all of the rest of them which it infects. But VSV infects also all kinds of cells in cell culture. So it's a very, very useful virus. And in fact, it's been used, and we'll talk about it later, for the current Ebola vaccine. So what people have done is taken one virus, in this case VSV, and then put some antigenic determinants on the outside of it. And that then has turned out to be a really useful source, not unlike, in fact, the Dengvaxia that Dr. Hirsch talked about when he talked about the flaviviruses, where they have a yellow fever backbone, or the parts of the virus that they then put extra pieces on top of. So this idea of having a Also, people call them pseudo-typed virus, and we'll talk about that a little bit in just a second, just having a receptor or something else on the outside of a different virus, which can cause a nice immune response, but then not cause disease. Also, partly because of that, VSV is also used in gene therapy as well, because it has this really broad host range. And so you can put whatever you want inside the genome of this virus, and it will deliver that to your favorite cell type. The Paramyxoviruses, these are the, well, I guess you could call rabies nasty as well. But particularly measles, mumps, rubella, these are all very similar in terms of their makeup, the way molecularly that they work, but are causing different diseases and, in fact, very, very widespread ones. So big difference, of course, between measles and rabies. Rabies basically have to get bit by some kind of rabid animal in order to get transmitted, whereas measles is transmitted in an airborne way. And that's partly this Paramyxoviruses, or myxoviruses are those which are being transported by the aerial mechanism. So measles, rinderpests, which is in italics here because this is one of the other huge successes that we've had in terms of eliminating disease. So most people have heard about smallpox that was completely eliminated. Rinderpest, which was the bovine equivalent of measles, has now been completely eliminated through vaccines and has been really very influential, particularly in the developing world, in terms of allowing a lot of these subsistence farmers to be able to survive. It was really nasty for cows, but through this vaccination process, you're able to completely get rid of it. So smallpox and rinderpest are really the only two that we've managed to get rid of so far. And polio, hopefully, some wood to knock on, will happen before too ridiculously long. Mumps, Nipah, and Hendra, these are other viruses, also these Paramyxoviruses, and then Sendai virus as well. These images, these are ones that are not from the textbook, but Viral Zone, I think I mentioned this earlier on in the course. These have these really nice diagrams, sort of cartoons of how all of these viruses form, but also a little bit of background on each of their replication processes as well. It's a nice complement, I think, to our textbook and the lectures. So here's one of these virions, has the proteins around the outside, but most importantly here we have our genome and the polymerase, which is associated with it. So again, the RDRP, RNA-dependent RNA polymerase, is always associated with the genome, and when that genome is released, that polymerase is going to be released at the same time. So let's look a bit more at these structures. If you just look at a TEM of the virions, they're pretty pleomorphic, so this probably looks a lot like the coronaviruses, only it doesn't have those big spikes sticking out of it, but otherwise these things kind of look like blobs. And that's why, again, I don't have a nice 3D printed image of these. They are classic enveloped viruses. Inside that envelope, most of these genomes are packaged as helices. Now, anybody remember, I don't know if Janan wants to toss the virus to someone, why helices are both really great ways to package genomes and really bad ways to package genomes? Not yet, because I've gotten to the bottom of the list. Let's see who catches it. So, why helical? On the nucleotides? Mm-hmm. Yeah, and that's one thing. The other thing is that with the helical symmetry, your identical interactions. Each of the subunits interacts with the subunit next to it in an absolutely identical way. You don't have to have quasi-equivalence where stuff is slightly different in terms of the interactions. But there's one other thing I'm thinking about in terms of helices that is different in terms of Toss the virion this side. Whoop. Uh-oh. We've got to work on our tossing here. Or something which is a little bit more aerodynamic. One and two. Pardon? Stability, yes, but what particular part of stability? Yeah, unwound when they're done, but so the great thing about a sphere is it's missing one particular thing. Or a circle. What does a circle not have? Ends. Helices have ends, and so those are not going to be protected. But if you're an enveloped virion, you've got the envelope around the outside, which is protecting it. And so that's probably why these are able to have these sort of helical symmetric genomes. And we'll see in excuse me, a little while later that this also is very important for how the genome is being replicated as well. Some of these, again, they've got these sort of blob shapes, and as we'll see when we talk about the filoviruses on Wednesday, these are very elongated virions, which also people will talk about them as being kind of filamentous. So these are, again, pretty blobby. The rhabdoviruses, people always used to think this is a completely different kind of virus because, you know, look, it's got this nicely defined structure, the bullet-shaped kind of structure. In terms of how they replicate, they're basically identical to each other. So there's something about these rhabdoviruses and the way that the proteins, and actually it's a little hard to see here, but the genome is literally right underneath the membrane here. So it's probably actually mostly the genome associated in a helical form, which ends up with these sort of rhabdovirus-like particles. But how you have one end that's different than the other, I think is still not actually known for any of these. And this is all a particular protein, which is associated with the genome and also associated with the membrane. It's a little bit easier to look at when we look at some cartoons. Here's the Paramyxovirus cartoon. You have the genome on the inside. Again, it's helical and packaged with a nucleocapsid protein, and we're going to see lots of these Ns and NC proteins in the next couple of lectures. So N, nucleocapsid or nuclear protein, the polymerase and a phosphoprotein, L and P. We'll see these in the genome in just a second here, but particularly this is that RNA-dependent RNA polymerase. So that's on the inside of the envelope. You also have a matrix protein. So we talked about the membrane protein in the coronaviruses. That's what's interacting between the nucleocapsid and the envelope proteins on the outside. And then the envelope proteins here contain a hemagglutinin neuraminidase. So this should sound, again, very, very familiar because hemagglutinins are binding red blood cells together and is usually what's going to be that receptor binding protein. Neuraminidase is chopping off that sialic acid to allow the virus particles to be released. In the Paramyxoviruses, there's actually a separate fusion protein. And so you have interaction with the receptor here, and then the fusion protein right next to it. These fusion proteins are going to fuse with the plasma membrane, so not endocytosis and structural changes there. It just seems to be pulling the virion close enough to a cell that then the fusion protein can function. That's good enough. So it's that binding. Once you have binding, then you're going to have fusion that takes place. The F protein has this fusion peptide. Again, it's hidden most of the time because once you've released a virion, you don't want to fuse back to the cell that you've just been infected from. Has a disulfide bond, but also is cleaved through proteolysis to generate this fusion peptide. And again, this is almost always at the end of the assembly process. And here's that hemagglutinin neuraminidase protein. The VSV does this slightly differently. And again, VSV is one of the rhabdovirus that people love to use as a model system. It actually has both the fusion protein and receptor binding combined in one protein called the G protein, not to be confused with GTPases, unfortunately. So the G protein here binds to the receptor and also gives you fusion at the plasma membrane. And so again, instead of having endocytosis or changes in terms of your pH, you end up getting a fusion that happens at the membrane. It's a little bit of endocytosis that happens, but it's mostly then fusion at the membranes. And because of that, what happens is you end up with just putting this G protein in, you can get cells to fuse to each other. And this also turns out to be really useful for gene therapy purposes. Again, you have your favorite gene inside one of these VSV, recombinant VSVs, to get it into a cell. All it has to have is this G protein on the outside. That will then, whatever you have on the inside will fuse to and release whatever genome piece you have on the inside. So lots of people use the VSV G protein on the outside of whatever they happen to have made in order to get gene fusion, fusion, and then their genes from the inside released inside the cell. And this is often what's called pseudotyping. So the pseudotype type is going to be what cells are normally going to be infected by a particular virus. If you now switch out that receptor binding protein, you can get a virus to bind to and potentially release its genome into a completely different cell. So pseudotyping is something that's just done quite a lot. And again, all this is mixing and matching these various different receptors. So once you get the genome released inside the cell, it's a helical nucleocapsid. And again, here we have our nice helical structure, each of these subunits all interacting with the one next to it in exactly the same way and interacting with the nucleic acid. What's interesting here is that the nucleic acid is not on the inside of the helix, like it was with tobacco mosaic virus, and we were talking about that before. It's actually on the outside of the helix. And this appears to be really important for protecting that RNA from getting degraded once it gets inside the cell. So this negative strand, so negative strand means that you're starting with the 3' end here. It's going to have these N proteins associated with it, and it turns out that each of these proteins binds to 6 and exactly 6 nucleotides. And in that process, will protect those nucleotides from getting degraded. And what it does is it binds on the backbone side, so where you have the phosphates and the riboses, and has the bases sticking out. And so you have the genome being released in the inside of the cell. The bases are already sticking out. And so what that means is that the RNA-dependent RNA polymerase can actually now bind to this RNA, even though it's associated with the nucleocapsid, and start to make new RNAs. Yeah?
Does this mean there's no replication or anything happening to open up the helix?
Yeah, so the question here is, so you wouldn't need an RNA helicase in this case, because just the fact that it's bound to the nucleocapsid protein means that the bases are all available. So they're not forming secondary structures. They're in this helix with the bases facing out and being available for the RNA-dependent RNA polymerase to bind to it. So this looks like the slide that I should have had before. I must have gotten them slightly out of order here. So where do you get fusion and how do they bind? Always going to be these membranes and plasma membranes. Many of them bind to sialic acid. Sialic acid, again, is the same one that flu is going to bind to. Classic hemagglutination outside of red blood cells form together. Measles binds to immune cells in particular. This is partly why measles is such a nasty disease, because it knocks down your immune system. And actually, having measles infections also can lead to other infections taking place. So measles is particularly bad, yet another reason to vaccinate. And the process is, your hemagglutination, you have binding, and then your F protein is going to change. These are classic trimers. Again, we've talked about this kind of ad nauseum in terms of how you get trimers. Those stick the fusion peptide into the membrane of the host. You have conformational changes that pull all of them together. So let's look at this genome now. Remember, it's packaged in this helix with the bases facing out and the nucleocapsid on the inside. So always packaged, also has the L and P proteins, which are the RNA-dependent RNA polymerases, bound to the 3' end of the genome. Remember, these are negative strands. So the beginning of the genome, first thing that comes inside the cell is going to be this leader sequence. This is where the RNA-dependent RNA polymerase is sitting. So reverse of what we had before again. They're negative strand viruses. So the genome is set up in such a way where you have N protein, PCV protein, M protein, F protein, HN protein. These are fused again in VSV to be a G protein. Again, the fusion and the binding proteins are all the same. So here we've got our structural proteins and our non-structural protein. And basically, the only real major non-structural protein is the RNA-dependent RNA polymerase down here at the end. So this particular version is Sendai virus, but measles, mumps, all of them are very, very similar here. So that's the coding sequence. But again, it's just one RNA. Has a small leader sequence, a small trailer sequence. Again, these are the non-coding sequences. But what's really most interesting is what's going on between each of these coding sequences. The gray region here. But what we have is the stop codon for the N protein, and it turns out it's the same for all of these other ones. Little bit of stretch of U residues, and I know those U's are going to turn into what? Yeah, exactly. Those are going to pair with the adenines. And then short sequence here, and then the start for your next transcript right here, very close to that. And so this process, these are about 30 nucleotides. How long are poly A tails usually? Who's up? Who's got the virion? How long are poly A tails? Are they 5 nucleotides? Yeah, 150, 200 usually. So there's not enough space between each of these guys, between each of these genes, in order to get your messenger RNA. So what happens? You have your RNA-dependent RNA polymerase that comes down, binds at the 3' end, will make a messenger RNA until it gets to this intergenic sequence. Sorry, this guy up here, get rid of that. When it gets to that intergenic sequence, it will make a whole bunch of A's. And so it just sits there on those U's and copies them until you get 150, 200 A's. And then, in many cases, this polymerase will actually fall off and go back to the beginning, start again, make a bunch more of this protein here, this RNA, get to the end, stutter for a while, and then in a few cases, it will start up again and make this next RNA, get to this next sequence, stutter a little bit, and then most of the times fall off and continue. And what that means is eventually you end up with, my pointer has stopped here, end up with much more of this RNA, the nRNA, and then these proteins that we'll talk about in just a second, M protein, F protein, HN protein, and the least amount of your polymerase. So how do you end up with more of the structural proteins? It's just because they're present at the 3' end of the genome. The polymerase gets to a certain point, will stutter a little bit, fall off most of the time, some of the time keep going, and just end up with more of the N protein, a little less of these PCV RNAs, less of the M protein, less of the F, less of the HN, and finally least of L. Yeah, Echo.
What is signaling it to do the stuttering? Like, how does it stop?
How does it stop stuttering?
Or how does it, why does it stay there?
Okay, why does it stay there? So why it's staying there, well, we'll get back to this in just a second here, and I think you're sort of realizing what we're just about to have a problem. But it just seems that whenever it hits this sequence, the polymerase itself will sit there and stutter for a while, and then eventually move on. And that just seems to be pretty random. And it's also true if you think about poly-A polymerases. 150 to 200 nucleotides. So eventually it will fall off or keep going. And so the length of those poly-A tails just seems to be relatively random, but at least it ends up with multiple copies of them. Okay, the other thing, which, and this is discussed in fact, quite nicely in this TWIV episode, if you make mutations in one of these open reading frames that causes the RNA-dependent polymerase to stop, then you don't get anything down here. And so what that means is that the polymerase isn't coming in at each of these intergenic segments and starting. It's staying on the template in order to make more of these. Yeah, John?
Is there any advantage to
Yeah, so what's the advantage to having the stuttering? Probably the advantage is you have a smaller genome. And so you don't have to code for all of these poly-A tails. And if you think about it, you'd have, a couple hundred in a poly-A tail here, 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 et cetera. So that's probably why. But again, why questions in biology, the answer is always what? Well, it works and, evolution. So yeah, there's no really good answer for that. It works and that's what seems to happen. So what's the obvious problem that I think that Echo was sort of driving at here? So this works great. Oops. Yeah. Except for now, I'm deciding not to work at all here. Okay. So much for the technology working. Go back to this one. Also not going to work. Oh, okay. So how do you get, so this is great for getting your transcripts, right? You get all your messenger RNAs, you got nice poly-A tails, it's good, it's wonderful, they get translated. But your genome doesn't have all of these long stretches of U's in it because we just decided that the genome would be too big if we had these hundreds of nucleotides there. So how do you get differences between transcript formation and genome formation? And so that's all about the concentration of the N protein. So at low concentrations of N protein, you stutter, and this may also be getting back to your question again, and at high concentration of N protein, you stop stuttering, and you will just copy the whole genome without making all of these stutters. So another question that you might have come up with is how do you make caps? Turns out that the L protein, also the RNA-dependent RNA polymerase, has a capping activity. All of this stuff is happening in the cytoplasm, so caps and tails are all being made by that polymerase. This is just another way of looking at this. Cartoon-wise, start out, you've got your genome that comes inside the cell. RNA-dependent RNA polymerase is sitting down at the 3' end. Will make your N messenger RNA stutter, piece of eRNA stutter, mRNA stutter, fRNA, HN, and L protein. This N messenger RNA is going to accumulate because you make the most of it. You're going to start making more and more N protein. At high N protein, then you switch from doing this kind of transcript making to making first antigenome and then genomic RNA. So I think it's a really smart, again, to totally over-anthropomorphize the process here. You have your N messenger RNA that gets made right at the beginning. You make lots and lots of that. And then when you have enough of that, you then start making genome. And then when you've made enough genome, then you're going to end up budding your way outside of the cell. So that hopefully is clear in terms of how we're getting genome formation versus messenger RNA formation. You may be confused, however, about this PCV thing. So why are we calling it PCV? What's that all about? Well, this gets to one of the other aspects that we have of these viruses, And that is that they, in a manner very similar to Phi-X174, can use different open reading frames to generate different proteins. The main protein which is made is the P protein, the phosphoprotein, part of the RNA-dependent RNA polymerase process, but you also make the C protein and the V protein, which are involved in basically messing with host defenses. And we'll get into any more detail about this as well. So there are a couple of different ways that you make different proteins. The first one is just by making, or starting, I should say, at a different place. So the normal start codon for the P protein is right here. But occasionally, the ribosome will start here, can also start here, can start here, or can start here. And some of these, so the P protein up here, is in the same reading frame as the V protein, but it's a different reading frame from the C protein. So 104 to 114 in nucleotides. Since it's 9 nucleotides would be in the same frame, 10 is off. So this generates a completely different protein. Turns out that these two are in the same reading frame, 81 and 114, it's a multiple of 3. And then another difference happens in these, and that is still when you're making the messenger RNA, the RNA polymerase, so the RNA-dependent RNA polymerase, gets to this run of, it's actually C's in the template, and occasionally will add a couple of extra nucleotides. And this seems to be exactly the same process that happens in adding the poly A tails, only it's a much shorter one, adds 1 or 2 nucleotides. And in that process, just changes the C terminus of the protein. So instead of having a long C terminus, which you have from the phosphoprotein, the V protein has a alternative C terminus and then hits a stop codon right here. So basically, there are two different things here. One is where your translation starts, and that's your alternative start codon. And the second one is some stuttering that happens not in an intergenic sequence, but an intragenic sequence. We end up changing the open reading frame and gives you a different protein. Aren't these amazing? Isn't evolution cool? So these are the processes where you're getting all these different proteins. We mentioned this genome replication already. How do they get out? This is packaging at the plasma membrane. Just buds. You have matrix protein together with your hemagglutinin neuraminidase. These then bud out from the cell. They are then released and can go off and infect some other cell. One thing that can happen here, particularly with these kinds of viruses, remember, they're fusing at the plasma membrane, then they're being produced at the plasma membrane. If there's a cell right next to it, very often you actually end up with the cells that end up fusing to each other. So basically, the virion buds, and it buds like straight into the next cell right next to it. And this is what's called a syncytia. And you can actually get this syncytia to happen by just making a bunch of the G protein. Remember, I mentioned the G protein is the one that's got the fusion and the binding specificity all in one. And so that syncytial formation can end up with multinucleate cells. And part of the reason that I spent a while talking about this is that these are the same kind of proteins which are also involved in placental development for placental mammals that probably came from an ancestral virus infection 300 million years ago. So cell fusion is almost definitely a viral process. Great for, getting your genome from one cell to the next, but it's a useful thing. Why don't the actual hosts decide that it's something that's useful that they can use? And I forgot to mention this before, but they mentioned the 6 nucleotides each that you have for the binding of the N protein. That means that the genome actually needs to be a multiple of 6 nucleotides, because if it isn't, you're going to have an end sticking out. And that end is going to be then very susceptible to nucleases, exonucleases inside the cell. So it's another reason that you can't have the variable length of these U residues to be able to put poly A tails on. And having that very specific sequence means that you're protecting the ends. And what that means is, again, if you're thinking about the polymerase, which can stutter, it can stutter just fine in the absence of the N protein, but as soon as it starts stuttering with your full-length genome, you're going to end up with ends of genomes that are going to get chewed up. So stuttering's great for making poly A tails, but stuttering's a real drag if you're going to be dependent on having this exact 6 nucleotide piece for packaging your genomes. Happy? Not happy? Ready for a clicker question? Okay, this should look very, very familiar, at least the questions, because I love to not have to come up with extra questions. So the structural proteins in measles are encoded at the 5' end of the genome, the 3' end of the genome, both ends of the genome, middle of the genome, spread throughout the genome. Ten, 5, we have consensus, at least close enough. Yes, again, 3' end of the genome. So we flipped them around relative to the coronaviruses. The same answer that we had before, and it's not C, like Stedman always does for his C answers. So is that clear? 3' end of the genome. That's where the RNA-dependent RNA polymerase is. Does the start-stop from that point on. So I did want to spend just a couple of minutes talking about disease, particularly as it's somewhat topical these days. Measles, respiratory disease. One of the things that I always like to do is show pictures of the disease rather than the vast majority of mainstream media that just show needles going into kids' arms. This is what you're preventing by having vaccines. Measles is a respiratory disease. It's about 14 days between when you're exposed to measles and you start to come down with a rash. That's why people are quarantined for 14 days after they've been exposed to someone who has measles. First thing that happens is you have a really high fever, and so that's the immediate diagnostics. One of the big problems with measles is that you're infectious 4 days before the rash actually comes up, and also 4 days after, but usually it's pretty obvious at that point that someone will be infectious. Part of that actually probably has to do with the fact that these are infecting immune cells, and those immune cells actually don't have a chance to form the nice reaction until later because they're being infected by the measles virus. The other big problem with measles is that unlike rabies, where you need to get bitten by the rabid animal, measles viruses can last in the environment for up to a couple of hours. And so someone could have been in this room 2 classes previous, and you could then get infected by the measles that was still here. So what do you do about it? Yes, vaccinate, 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 vaccinate. So why? Well, first one is that between 0.1 and 0.2% of kids that get measles die. Mostly in the developing world. That's not so much of an issue here in the US. But even here, in the developed world, between 1 and 2 in 1,000 have encephalitis, which is swelling of the brain leading to deafness, intellectual disabilities, etc. So 1 in 1,000, I think, is pretty bad. Pneumonia, which is the main cause, particularly in the developing world, on average, about 1 in 250,000 kids who get vaccinated have serious side effects. So 0.0004% versus 0.1 to 0.2%. So some kind of allergic effect, 2,000 times less likely than death. I like those odds. I'm going to get vaccinated. Before vaccination in the US, there are about 4 million cases of measles every year, 1,000 disabled per year due to this encephalitis. And measles was eliminated, quote unquote, in the US in 2000, and that's just because it wasn't spreading here. Why? Let's see. I know correlation is not causation, but I think these are pretty striking data. Thousands of cases of measles. Here's where the vaccine comes in. Boom. We go down to many, many fewer cases of measles. And this has been shown in lots of other places as well. Still had a few thousand cases every year till a second dose. This is now blowing up this region right here. And we've gone to practically nothing in terms of disease, with some exceptions, again, that we'll get back to in just a second. Just because there's no disease in the US, or very little disease in the US, doesn't mean that there's still not a problem with measles in the rest of the world. In 2000, there were almost 500,000 deaths from measles, and that was when about 72% of children were vaccinated. In 2016, we've reduced that to under 100,000, but there are still literally millions of people who get infected every year. So what does that look like on a global map? This is the measles incidence per million people over the last 12 months, really concentrated in sub-Saharan Africa, but also a few other cases. The Ukraine has a very high rate of measles. Madagascar has an incredibly high rate of measles. But even places like India, Pakistan, Philippines, very, very high rates of measles. And so what's happening with the measles in the rest of the world, and why are we still seeing it in the US? Because people go to places and they come back 4 days before anyone sees any symptoms. These things last for hours in the environment. They are going to get moved to the US or wherever you happen to be. Partly because we have a pretty high population, number of reported cases in the last 6 months. You can see that the US is actually getting up there in terms of actual numbers of recorded measles cases. Why is that? This is one of my favorite tweets ever. I will let you read it, but something that I retweet on a regular basis. Why is this scary? Well, these are the data, I should say, for this year. 704 measles cases as of last Monday. It's probably about 720 by now in 2019. So we're pushing the point. Remember what I said about 1 in 500 to 1 in 1,000? We're going to start having some really nasty disease and deaths in the US from measles, because people aren't vaccinating. So why don't we leave it on that wonderful, happy note, and then we'll talk about things that people seem to be much more scared about, namely Ebola and Marburg, on Wednesday.